Hi everybody. So when we go back to classes and some of the topics we were covering recently, one of the ones we looked at was the polymerase chain reaction, and it's known as PCR. It's probably the technique that transformed molecular biology and added so much, I suppose, new applications, how you could use molecular biology in different, I suppose, scenarios and situations. So what I wanted to do here was, obviously, if you look at this slide, I want to talk a little bit about how it was invented and its use, its mechanism of action, and so that you'd be able to explain and maybe train people on the technique. If you can explain it, it means you understand it. And I want you to be aware of controls, negative and positive controls in this technique. So what we did was I provided you with a pre-practical video on PCR, and I would highly suggest, before you keep going with this video, to make sure you've watched that. I'm going to link it here and in right about here, and I'm also going to link it in the description underneath the video in YouTube, so you can click on the link if you haven't seen it already. It's a short video that just highlights through an animation how the polymerase chain reaction works. So I'd like you to go back and watch that video and then come back to this video now. If you've seen it already, well, let's go. So now you know that this technique involves three main stages, and the technique involves those stages known as denaturation, annealing, and extension. So when we add excess heat, our DNA is going to be separated because the hydrogen bonds get broken. We are then going to use an annealing temperature where we lower the temperature, and it's a temperature approximately 55. It's very dependent on the actual primers that are being used in the reaction. But those primers will find their complementary sequence, as you can see here and here. And once they've found their sequence, we're able to change the temperature again to something called the extension temperature. And during the extension phase at 72 degrees, there's an enzyme called TAC DNA polymerase. So we've met DNA polymerases before in replication. They extended primers on the 3' end, and now we're meeting one that's called TAC, TAQ, and that stands for the species where we obtain that enzyme from. It's a thermostable Thermus aquaticus bacteria. So by getting that enzyme, it allows the enzyme to survive in these high temperatures, 95, 55, 72, back again, 95. Because we do these three steps actually about 30 to 35 times in normal PCR, and they can be as high as 40 to 50 times in other applications of PCR that we'll discuss shortly. So it's very important to realize why we need a thermostable DNA polymerase, and it's known as TAC DNA polymerase. So by going through this step once, can you see at the bottom of this diagram here, we now have a copy of what was at the top. So at the top we had these two yellow sequences that were known as the target sequence. On the sides of the target sequence we had these flanking regions where our primers were going to anneal to, and then we were going to extend. Well, as we keep going through these, say these cycles, in the first one, we've kind of looked at there the denature, anneal, and extend. As we progress and do this more and more and more, we're going to start generating just the target regions. And over a case of 30 to 35 cycles, you actually start increasing exponentially. So in a couple of hours, we can turn, for example, one target region in a source into millions of copies of just that target region. So ultimately, through subsequent cycles, we'll have made amplified numbers of just the target sequence. So this has so much potential. We're hearing it about at the moment in our applications with regards to diagnostics. So at the moment, looking for a virus to see if it's present or absent in a particular sample, we only need a very small starting material. And because PCR can amplify the target region, we can then find out if something is present or not. We can bring it up to levels that are detectable. It can be used in, say, forensics and so on. We talked about a lot of these in class before we finished up. So PCR can almost give us the DNA region that we want from very small starting material. So if you wanted, say, the insulin gene, in our last sessions in this handout, we talked about recombinant DNA technology and getting living systems to make therapeutics, such as in the biopharma industry. And if we want the insulin gene to be able to put into a plasmid with restriction enzymes and then put into a particular expression system, such as a bacteria, or in higher level proteins, we'd need a mammalian cell. But if we wanted that piece of DNA, well, where do we get it from? And what we can do is design specific primers against the target region. And we decide what region gets amplified by the primers that we design. So, for example, these primers will be designed against insulin target regions. One will bind to one strand and one is designed to go to the other strand. So every PCR has two primers involved. They call them a forward and a reverse primer. And those primers are going to amplify just the target region. So if we do that, we can get millions and millions of copies of, say, the insulin gene, and then we can use that gene to try and get it into an expression system to make insulin for a therapeutic. So what we can do, even with our detection, if I gave you, say, cells in culture and asked you to tell me was a particular bacteria contaminating that liquid media, well, if you have primers that are designed against the target region, so imagine these primers here were designed against the bacterial DNA that you want to see is it contaminating the sample. Well, let's say there was bacteria there. Well, these primers will find their target and hybridize by hydrogen bonding to their target. And then when the Taq polymerase is present, we will extend these primers on the 3' end and amplify the product. We'll be able to detect that product when it's at higher levels, higher numbers, and then we know the bacteria was obviously present. Where if the bacteria is not present, the primers have nothing to bind to. They cannot find the target region that was the bacterial DNA. The primers don't anneal, and that way we have no extension and no product detected. So currently at the moment, PCR, or a version of PCR, is being used to detect for the presence or absence of a virus in all these samples that are being taken from people. So if the virus is present, we'll see the primers anneal to that viral sequence and we'll see an amplification event occur. Where if the virus is not present, those primers don't find its target region and no amplification occurs. So I'd advise you, as I said, to watch over that video that I had prepared for the lab to give you an idea of PCR. Common to PCR in general, I want to kind of, I suppose, get you to start thinking about controls. And the reason with controls is I think they're vital. You cannot trust any data or any experiment unless there's an associated control. I think I would often have heard before, you're only as good as your controls that you have in an experiment. So what does a control do? Well, in general, it validates, it verifies, and it trusts your data. And they're three very, very important things in science with regards to integrity of your work and making sure you can stand over your data and the outcomes. So ultimately in science, you have to be able to defend your work and prove that it's correct. And you can stand over and believe and trust and validate all the results. And you need controls in order to do that. So I'm sure in lots of experiments that you've done so far in the last few years, you've always included a control. We used ones before that were, no enzyme controls or no substrate controls and so on. So there's all these different types of controls. So if we have to validate, verify and trust, I want you to think of this exercise. So I want you to design in your head now a PCR diagnostic lab. Okay, so I want you to consider that you are that test that you're going to diagnose a presence or absence, in my example here, a bacteria in a food sample. So you will have primers designed against that particular bacterial DNA. And if the primers can detect, can find, locate, hybridize to the bacterial DNA, they'll amplify and give us a product that we can detect on, an agarose gel if we do normal PCR. So I want us to think about the controls for a second. So I want us to think about negative and positive controls that we could put in our experiment to make sure everything is going to work okay. So in general, PCR results are put on a gel, and I want us to draw kind of almost the gel results as to what we'd expect. So what I'd advise you to do here is, as we go through, we'll press pause on the video, maybe give a sketch, take a few minutes, and then you can come back to the video to see what we did. So let's say, for example, what would it look like if we had a contaminated food? So this was a food that has a particular bacteria being detected. What would a gel look like if it was a non-contaminated food? And finally, what would it look like if all our reagents were actually contaminated? So that's something that we have to really consider with PCR, because if we give a false result to somebody in any of these detection methods, that can have a catastrophic event. So we have catastrophic for the individual. And again, integrity, your validation process has to be tested, and the integrity has to be there with all your results. So the target region, let's say, for example, in the bacteria that we want to amplify is going to be 300 base pairs. So we design a forward primer and a reverse primer, and they will hybridize to their complementary sequence. So if the bacterial DNA is present, the primers will hybridize, they will be extended, and will amplify just the target region, which now we know is 300 base pairs in size. So let's think of our gel layout. Okay, this is our sample gel. So I want you to review what's going to be in every lane. This is our marker, 100, 200, say this is 300 base pairs in the marker. Marker for in lane 1. The second one is food sample A and food sample B. So these could be duplicates of the same food sample, just taken that you do a duplicate of the experiment. For example, sometimes in a company I worked in before, they would test the sample in one office, and then equally the sample would be tested by a different person in another office on another part of the planet, and then the results had to obviously be the same. But it was really making sure they had a duplicate, independent verification of the result. Let's say lane 4 is a positive bacteria sample. So this is a sample with just the bacteria in the tube. It's one to make sure that your primers are working, that your enzyme is working, and that it amplifies the target region as you'd expect. And then in lane 5, we're going to do something called an NTC, a no template control, NTC. So what happens here is, instead of putting in a food sample to test or a positive control or anything like that, we just put in sterile water instead of the template. So here the primers should find nothing to amplify. It's a no template control. There's no template for the primers to anneal to, so no extension should take place, and ultimately no product and no amplification. So it's kind of like an empty control. If we do see any amplification in lane 5 in the negative control, or this NTC, well, then we know there's something wrong. The primers have found their target somewhere, and we must have contamination in one of the reagents. And that's one of the things we have to consider. So what I want you to do now is I want you to do a gel out on a piece of paper, like what we have here. And I want you to go back and look at scenario 1, where we have a contaminated food result. What would it look like on this gel? Where would you draw the bands on the gel, the lines that will be amplified if the target region is, or the lines that will be present if the target region is amplified? I want you to draw the gel again and consider a non-contaminated food result. So a food result where it's a negative, that bacteria isn't present. And then finally, I want you to do scenario 3, where we have reagent contamination. So there's 3 scenarios I want you to do with this gel. So draw this out 3 times, sketch it out on a piece of paper, and go back to your 3 scenarios and draw out exactly what you'd expect in each lane for those 3 cases, bearing in mind that the target region being amplified is 300 base pairs. Right? That's where the primers are designed, 300 base pairs away from each other, and that's the size that'll be amplified if the target is detected. Okay, so please press pause, give that a go, and come back and press play in a few moments' time. Okay, so hopefully you've gone through that exercise, because it'll really help you understand the idea of controls. And let's see, does my results match what you have on your piece of paper in front of you? So the food contains the bacteria. So what happened here? Lane 1, we have our marker. Lane 2, we have a band at 300 base pairs. 3, a band at 300 base pairs. 4, a band at 300 base pairs. And 5, we have nothing. So what's happened? Let's check number 5. 5 is our no template control. So there was sterile water, there was no food, no bacteria, no template for it to amplify. And in this case, the primers inside in the reaction didn't anneal to anything because there was no template. So that means that none of our reagents are contaminated, and our result is going to be quite valid with regards to no contamination. 4 was the positive control. So it's a positive bacteria. We give it the DNA. We should see amplification. We do. If we didn't see anything here, you could blame the enzyme. Maybe the enzyme's not working. And then food sample A and B, we have detection of that 300 base pair band. So we know now that the bacteria was amplified in the food sample, and the food does have this contamination bacteria. Second scenario, the food does not contain the bacteria. So let's start again with the negative control, the NTC. It's empty, so that means that we have no amplification in our NTC, so our reagents are okay. Let's check our positive bacteria sample. We see amplification as we expect in the positive control at 300 base pairs, and in our duplicate food samples in lanes 2 and 3, we see no amplification. So can we stand over this result? Well, our positive is working, so we know our enzyme and our machine is working okay. Our NTC shows no amplification, so we know our reagents are okay. And in lanes 2 and 3, we know there's no amplification of our product, so we know the food does not contain the bacteria. The third scenario was if we had reagent contamination and something called an invalid PCR. So again, let's start with the no template control in lane 5, and now we have amplification. So remember, this was a sample, a tube that we set up with the enzyme, with the buffer, with the primers and sterile water. So the primers shouldn't find anything. And in this lane, they have amplified a 300 base pair product. So that tells us that the primers did find a target region and did amplify. And that's a bit of a problem. It informs us that we have amplification in our NTC and our reagents have to be contaminated, be it the buffer, the primers, the water, something is contaminated and that's causing an issue. So do I even look at the other lanes? No, I have no interest in analysing it further, because if I don't have faith in one of my reagents, I can't stand over whether the food is or is not contaminated, because I don't know if it's coming from just a reagent. And that's something I have to go back to the drawing board, replace all my reagents and repeat the experiment again. So I hope that little example gave you a nice work through of the importance of controls and how vital it is to do it. So let's just recap what we've talked about so far. PCR is an incredible technique developed in the 80s by a scientist called Kary Mullis. It involves a thermostable DNA polymerase that's able to amplify target regions by going through particular cycles of 3 stages: denature, anneal, extend. And we go through those cycles about 30 to 35 times in normal PCR, or 40 to 50 in other applications that we'll talk about soon. PCR is able to locate target regions and amplify just those target regions. So a key aspect that I really want you to gather and understand is what determines the target region that's going to be amplified. Be it insulin, if you wanted the insulin piece of DNA, be it the bacteria that we detected in the food sample here in this control experiment, or be it the idea of detecting a virus in a sample. What dictates that? So going back to the diagram we looked at, I want you to really grasp the idea that it's the primers. It's the primers that you design. They're the idea. They're the reagents that specify what will be amplified. All you have to do is design a primer online, upload the sequence you want, and a company within a few days' time will have sent you the primer for your PCR reaction. And that primer has to be complementary to its target region. We use primers in sets of 2, forward and reverse, but that's what specifies the target region to be amplified. We've also looked at controls. And we've looked at scenarios with this example of validating, verifying, trusting our data. And I've talked you through these 3 gel systems, showing us 3 different scenarios. And I think that's hopefully helping you a little bit with the concept of how a control can be used. Okay, so that's a nice summary of what we've done so far. The other slides that we discussed in class, I know you're probably saying I'm flicking through them, but we would have looked through a lot of these applications in our class. That it can be used for diagnostics. You can see if something is present or absent. We only need tiny amounts. So, for example, if there were only 10 bacteria per million human cells, we can detect that with PCR. So think of forensics, where we have very low starting material. We can really detect it. Think about virus detection. If we want to find out if someone is at an early phase where the titer or the amount of virus present is low or the amount of virus present is high, we only need tiny amounts that we can be able to detect this. We don't have to wait until day 40 before we can detect things. We can detect them really early. So PCR is something where it has so many applications. It really is endless. It's used a lot in different cases. Like even here we talk about someone's HLA type. It's the where we're looking to see our, say, transplant organs compatible between the donor and the recipient? Will they match? It's often used in forensics. It's been used in parentage in disputed paternity cases or immigration cases, semen samples, implying guilt or innocence in numerous assault and rape cases, and just even a single hair, the root of a single hair, gives us enough DNA that we could PCR, and when we have enough of it, we can determine the sequence. We can actually get it sequenced and find out the exact composition of that piece of DNA. DNA is very stable, so it means that even if we have samples that might be, I suppose, as long as they're preserved in some way, but they can last a lot of time, and that's something that lets us, work with pieces of DNA that can be quite old and still determine an outcome from that. So I guess what I want you to consider is now, what all this PCR, what could we use it for, and how can it be, I suppose, built into, you know about detection, you know about controls, you know about this technique, but how can we bring it even further? How has the technique developed from the very early stages, other formats of it? And that's where we want to go now and talk about other applications of PCR. Okay, so for example, the names of these techniques, I suppose one of them we're going to talk about first is called reverse transcription PCR. And then once we've gone through reverse transcription, we're going to move into something called real-time or quantitative PCR. And that's kind of what we were discussing towards the end. So I hope that this video has given you, I suppose, a nice overview of PCR and gives you a view of where we're going to go with it shortly. Okay, talk to you soon.